adult human hemoglobin is hemoglobin A, which is composed of 2 alpha globin chains and 2 beta globin chains. It's a tetramer of 2 alpha and 2 beta globin chains, as you can see in this picture. Alpha globin gene is located in chromosome 16, whereas the beta globin gene is located in chromosome 11. Due to various mutations in these genes, the mutations in alpha globin gene leads to reduced or absent production of alpha globin chains that leads to alpha thalassemia. Similarly, the mutations in the beta globin gene leads to reduced or absent production of beta globin chains in erythroid cells in humans, which leads to beta thalassemia. If we briefly look at the molecular basis of alpha thalassemia, alpha globin gene is a duplicated gene. That means there are 2 copies of alpha globin gene in each chromosome. So in a cell, there are 4 copies of alpha globin genes. Mutation in 1 or 2 alpha globin genes, as you can see in these 2 situations, leads to an asymptomatic state, which is known as alpha thalassemia trait. Mutation in 3 out of 4 alpha globin genes leads to hemoglobin H disease, which is a sort of a milder disease than beta thalassemia major and a non-transfusion thalassemia, non-transfusion dependent thalassemia. In situations where you have all 4 alpha globin genes mutated, there will not be any alpha globin produced. As we all know, alpha globin is a component of fetal hemoglobin, which is hemoglobin F. So even the fetus does not produce hemoglobin. This condition leads to what known as hemoglobin Barrett's hydroxyurea, and it is invariably fatal in utero. This is the only slide that I'm going to discuss about alpha thalassemia, because the predominant problem we see in our country is beta thalassemia. This diagram shows the genetic basis of beta thalassemia. As you can see in this, as opposed to alpha thalassemia, in a particular chromosome, there will only be 1 beta globin gene. So in a cell, there will be 2 beta globin genes. Beta globin genes behaves as a normal sort of a autosomal recessively inherited gene. Therefore, mutation in 1 gene does not lead to clinically significant problem. Mutation in 1 beta globin gene leads to asymptomatic anemia, which is known as beta thalassemia trait. When you have mutations in both the beta globin genes, that leads to a severe clinically significant anemia, which is known as beta thalassemia major. When we discuss about beta thalassemia, it is important to identify a specific subset of beta thalassemia, which is known as hemoglobin E disease. Hemoglobin E is a specific point mutation in one of the codons of the beta globin gene. The problem with this mutation is hemoglobin E is a structurally abnormal beta globin. It produces a structurally abnormal beta globin. At the same time, it is produced at reduced rate. So hemoglobin E or E mutation behaves as a thalassemia mutation. However, the reduction in beta globin production in E mutation is relatively less severe than in beta thalassemia mutation. Therefore, individuals with heterozygous mutations with hemoglobin E mutation will again be asymptomatic and has the disease known as beta hemoglobin E trait. Similarly, this hemoglobin E can be inherited homozygously. As opposed to beta thalassemia major, homozygous hemoglobin E disease is a milder disease, and most patients will not require transfusion because there will be sufficient amount of hemoglobin E produced to maintain the hemoglobin. However, when E mutation is co-inherited with the beta thalassemia mutation, the condition which is known as hemoglobin E beta thalassemia, that behaves similar to beta thalassemia major, more or less similar to beta thalassemia major. Therefore, the clinically significant beta thalassemia syndromes that we see in clinical practice are homozygous beta thalassemia major and hemoglobin E beta thalassemia. Beta thalassemia is possibly the most common genetic disorder in the world, and it is estimated that 70,000 children are born each year with beta thalassemia. Most of these births occur in the traditional thalassemia belt, which is a tropical belt which extends from the Mediterranean region, Sub-Saharan Africa, Middle East to South and Southeast Asia. As you can clearly see, Sri Lanka is within the beta thalassemia belt. The situation in Sri Lanka is shown in this figure. Beta thalassemia carrier frequency in Sri Lanka is reported as 2.8%. That means 1 in 40, approximately 1 in 40 individuals in Sri Lanka carries the beta thalassemia mutation. So they will be beta thalassemia trait. Approximately 60 children are born with thalassemia each year. This is beta thalassemia major or hemoglobin E beta thalassemia. And currently, we have about 1,800 patients who are being treated for beta thalassemia in 26 different centers island-wide. As you can see, most of these patients are receiving treatment at Kurunagala, Ragama, and Anuradhapura thalassemia centers. 68% of our thalassemia population has beta thalassemia major, that is homozygous mutation, and 20% has hemoglobin E beta thalassemia. The clinical classification of beta thalassemia is quite straightforward. It can be divided into transfusion dependent thalassemia and non-transfusion dependent thalassemia. Patients with transfusion-dependent thalassemia will present within the first 2 years of life. They will have marked bony changes, hepatosplenomegaly, and their hemoglobin will drop if they are not transfused. It will invariably gradually drop below 7, 6, 5 if you don't transfuse. This group composed of homozygous beta thalassemia patients, which are also known as beta thalassemia major. Hemoglobin E beta thalassemia, as you will see in the next few slides, is a spectrum of disorder. Some patients of hemoglobin E beta thalassemia will be classified as transfusion dependent thalassemia, which we will discuss a bit later. Non-transfusion dependent thalassemia patients are beta thalassemia intermedia patients, which is a complex genotype. It can be either generally they have heterozygous beta thalassemia mutation who would be otherwise beta thalassemia trait, but due to various genetic modifiers, they have a sort of more severe anemia than beta thalassemia trait, so they will require infrequent blood transfusion. Similarly, mild to moderate hemoglobin E beta thalassemia patients will be non-transfusion dependent thalassemia. These patients will generally present after 2 years of life and have milder anemia, and they will be able to maintain the baseline hemoglobin at around 6 to 7 grams per deciliter without transfusion. So, difference between transfusion dependent and non-transfusion dependent is if you don't transfuse, patients with transfusion-dependent thalassemia will drop their hemoglobin to a very low levels. It will not stay at 7, 6, 5, 3, it will drop. Whereas non-transfusion-dependent thalassemia patients, they will have anemia. Their hemoglobin will be somewhere between 6, 7, or 8. But if you don't transfuse, they will remain to have that baseline hemoglobin between 6, 7, and 8. Before we move on to the treatment of thalassemia, it is important that we understand the pathophysiology of thalassemia, because most therapies are based on the pathophysiology. As you can see in this picture, the predominant defect is in the beta globin gene. There will be reduced absence or reduced production of beta globin, which will lead to reduced hemoglobin A synthesis. However, the production of alpha globin continues normally. Some of these alpha globin can combine with gamma globin to produce hemoglobin F. But most alpha globin will be unpaired alpha globin, which will be an excess-free alpha globin, which precipitate in erythroid cells and causes destruction of these red cells in circulation, which is known as hemolysis, or in the bone marrow, ineffective erythropoiesis. Both these will lead to anemia. Due to anemia, these patients will receive multiple transfusions, and in patients with thalassemia, the intestinal iron absorption is increased. Both these will lead to iron overload. Iron overload will invariably lead to all the complications that we are familiar with. Current therapies of thalassemia basically address three of these sort of areas in the pathophysiology. Currently, most patients with thalassemia receive only supportive treatment with blood transfusions and iron chelation, as we know, and the only available curative treatment is hematopoietic stem cell transplantation, which is widely known as bone marrow transplantation. Before we move on to novel therapies, we'll briefly discuss the current therapies. Blood transfusion is the cornerstone or the mainstay of treatment, supportive treatment for beta thalassemia. In transfusion-dependent thalassemia patients, it is generally commenced around 6 months of age when the hemoglobin falls below 7 grams per deciliter. In these patients, transfusions are given to promote normal growth, allow normal physical activity, adequately suppress the bone marrow activity, and suppress extramedullary hematopoiesis to suppress the hepatosplenomegaly. These patients should be transfused with leukodepleted packed red blood cells, which are as fresh as possible, ideally less than 2 weeks old. The aim of transfusions in transfusion-dependent thalassemia patients is to maintain a pre-transfusion hemoglobin between 9 and 10 grams per deciliter. The way you do that is immaterial. If you can maintain a pre-transfusion hemoglobin between 9 to 10 grams per deciliter, then you will be managing these patients successfully. Generally, they will require blood transfusions 2 to 5 weeks. As opposed to transfusion-dependent thalassemia, the transfusion therapy for non-transfusion-dependent thalassemia is a bit different, and it's not very clear-cut. Therefore, many patients with non-transfusion-dependent thalassemia, especially patients with hemoglobin E beta thalassemia, will have certain problems. As I have already mentioned, these patients can maintain a baseline hemoglobin between 6 and 7 without transfusion. Therefore, need for transfusion should be decided individually. Transfusions are generally indicated if they are symptomatic, if they have growth failure, severe facial changes, or if they have a rapidly enlarging spleen. Hemoglobin per se should not be an indication for transfusion unless it is sort of, you can't really say a value, but generally in our practice, we will tolerate the hemoglobin between 6 and 7 if they can be managed without transfusions. This clearly shows the spectrum of hemoglobin E beta thalassemia, which is a non-transfusion dependent thalassemia. I want to speak few more words about hemoglobin E beta thalassemia. One, because there's no clear-cut guideline on the transfusion of these patients. Secondly, it seems that because we don't know exactly what to do, in this country, these patients have been managed with different regimes of transfusion. So it's important that we try to understand how best to transfuse these patients. So your hemoglobin E beta thalassemia patients can sit at any point of the spectrum. Some of these hemoglobin E beta thalassemia patients will never receive a transfusion. They will be completely asymptomatic and they will not require a transfusion. Then they will not be a problem. Certain proportion will require monthly regular transfusions because they have severe transfusion dependent thalassemia. And a large majority will be in between, who will be able to manage as non-transfusion dependent thalassemia because they maintain a hemoglobin between 6, 7 or 8. However, when we evaluated the transfusion regimes used for thalassemia patients, hemoglobin E thalassemia and beta thalassemia major, we saw some drastic sort of findings in this research. We recently evaluated the blood transfusion therapy for patients with thalassemia major and hemoglobin E beta thalassemia, which are labeled as transfusion dependent patients in Ragama, Anuradhapura, and Kurunegala thalassemia centers. As you can see in these graphs, patients with beta thalassemia major were maintaining significantly higher pre-transfusion hemoglobin levels compared to transfusion dependent hemoglobin E beta thalassemia. Here we have taken only the patients who have been managed as transfusion dependent hemoglobin E beta thalassemia. However, if you see this sort of a graph which shows annual transfusion requirements, it is clear that there is no difference between annual transfusion requirements in these two groups of patients. It seems that in this country, in most centers, we are providing same amount of blood to these patients, but at the same time, we are not maintaining the adequate pre-transfusion hemoglobin. So what's happening in the country, in thalassemia major patients, we will maintain a pre-transfusion hemoglobin above 9 and maybe transfuse up to 14 monthly. When it comes to hemoglobin E thalassemia patients, what's happening is you assume that they can tolerate a lower hemoglobin and you allow the hemoglobin to drop to maybe 6 or 7 and then still transfuse monthly up to about 11 or 12. So you are not sort of, if you can maintain a pre-transfusion hemoglobin between 6 or 7 without transfusion, that's fine. But if you are transfusing them, you need to manage them exactly in the same way that you would manage beta thalassemia major patient and transfuse them adequately to maintain a pre-transfusion hemoglobin above 9. I hope I made it clear. Right. So that's about blood transfusion. The iron chelation, I'm not going to talk much about iron chelation because we all know the 3 iron chelators that are available at the moment are deferoxyrox, desferrioxamine, and deferiprone. And I'm not going to spend time on this iron chelation. Bone marrow transplantation or hematopoietic stem cell transplantation is the only curative treatment option available for beta thalassemia, and approximately more than 3,000 bone marrow transplantations for thalassemia have been performed in the world. And in Sri Lanka, we have recently started bone marrow transplantations in the private sectors. Indication for bone marrow transplantation or hematopoietic stem cell transplantation in thalassemia is transfusion-dependent thalassemia. It has to be done as early as possible when it is feasible during first few years of life before iron-related complications appear. The problem with bone marrow transplantation is the lack of donors. Even if you have money, you will not be able to undergo a bone marrow transplantation if you don't have a suitable donor. The most sort of the best donor is HLA identical sibling donor. If you have an HLA matched sibling donor, survival is more than 90%. If you don't have an HLA matched sibling donor, the survival and the success rate of bone marrow transplantation drastically reduced and can range between 60 to 90%. So it becomes less successful if the donor is not an HLA matched sibling donor. Other possible donors that can be used for hematopoietic stem cell transplantation are HLA matched unrelated donors who are found from the donor registries, or HLA matched cord blood, or haploidentical related donors. That is, one of the parents. One of the parents will be half matched to patients with thalassemia. So parental donors can also be used. But I repeat, success rate with these donors are much less than using HLA identical sibling donors. Hematopoietic stem cell transplantation is a procedure with complications. There is transplant-related mortality, which can range between 5 to 10%. The patients can develop severe graft-versus-host disease, or sometimes the graft might be rejected. So you will go back to your thalassemia state. So that's about hematopoietic stem cell transplantation. Then for the next 20 minutes or so, we will move on to novel therapies which are in the development or which are sort of just approved for use in patients with thalassemia. These therapies are of two forms. They can be pharmacological therapies or the more advanced gene or cell-based therapies. And those therapies will also act at different places of the pathophysiology of thalassemia. Two pharmacological therapies which are sort of showing some promise are hydroxyurea and luspatercept. Hydroxyurea induces hemoglobin F, whereas luspatercept is an inhibitor of ineffective erythropoiesis. Gene therapy can be used to correct the abnormal beta globin gene as well as to address the alpha to beta globin imbalance by either inducing gamma globin or reducing the alpha globin synthesis. If we discuss one by one briefly, hydroxyurea is a cytotoxic anti-cancer drug which has been used to treat several malignancies for more than 3-4 decades. However, it also increases the hemoglobin F or fetal hemoglobin by reactivating gamma globin. The dose of hydroxyurea is 10 to 20 milligrams per kilogram per day, and it has been shown to be effective in patients with non-transfusion dependent thalassemia as well as in patients with sickle cell anemia. So most of the time it is used in certain non-transfusion dependent thalassemia patients. However, the effect of hydroxyurea, there's very little evidence to support the use of hydroxyurea in transfusion dependent thalassemia patients. Hydroxyurea has some side effects. Its tolerability is not great. The patients can develop GI discomfort, hyperpigmentation, or bone marrow suppression. Because the evidence is lacking for use of hydroxyurea in transfusion-dependent thalassemia patients, we recently started doing this randomized double-blind placebo-controlled clinical trial in Dragama thalassemia unit in adult patients with transfusion-dependent beta thalassemia. We have manufactured exactly similar identical placebo to hydroxyurea, and we have been doing this trial for the last 1.5 years. So what are the results that we have found? What have we found in this? These are unpublished preliminary data. The trial is still ongoing. I would say we have found mixed results. In certain patients, the results are not that promising, whereas in certain group, there is some evidence that hydroxyurea is beneficial. What we did was to randomize patients using random number charts to two groups to either take hydroxyurea or placebo, and the patients were treated for 6 months. So far, 38 of our patients have completed the trial, 18 hydroxyurea and 20 placebo patients. Of those 18 patients, 6 patients we identified as responders by evaluating their hemoglobin F. And these patients required 20% lower blood volume compared to control arm patients. At the same time, they maintained higher pre-transfusion hemoglobin. Importantly, we did not observe any major serious side effects due to hydroxyurea. So at the moment, we can say that it seems hydroxyurea is effective in a subset of patients with beta thalassemia, transfusion dependent thalassemia, but not fully effective. We are further evaluating the sort of confounders or the other factors that will predict the response to hydroxyurea in these patients. That's all about hydroxyurea. Then we will move on to the new activin 2 receptor trap drug which was developed, which is known as luspatercept. Growth differentiation factor level plays a critical role in the ineffective erythropoiesis, not only in thalassemia, in many other chronic hematological diseases. So what luspatercept does is to block the interaction between GDF11 and activin receptors. The phase 3 clinical trial using luspatercept in beta thalassemia major and transfusion dependent patients was recently concluded. This was done in Italy and published in New England Journal of Medicine. They used 336 patients, and in a randomized double-blind placebo-controlled way, they gave luspatercept subcutaneously 3 weekly. What they found was, again, similar to hydroxyurea, not fully effective only a subset of patients. So similar to our findings in hydroxyurea, they found it is effective in 44 out of 224 patients who received the drug, and they saw a reduction of transfusion burden by 33%, whereas we saw a reduction in transfusion burden of about 20% by using hydroxyurea. However, this trial is already published, and based on the findings of this trial, now luspatercept has been approved by the FDA, US FDA, to be used in patients, adult patients with transfusion-dependent thalassemia. Right, so those are the pharmacological therapies, novel pharmacological therapies that have shown certain promise. However, gene therapy has been in the sort of a arena for more than 2 decades. Beta thalassemia is one of the prime candidate diseases for gene therapy. What exactly does gene therapy mean? So we all know the problem with beta thalassemia is the beta globin gene is mutated. So the principle behind gene therapy is to introduce an exogenous normal beta globin gene to erythroid or hematopoietic stem cells of patients with thalassemia. So this cell has the defective gene. You introduce the healthy gene. You don't do anything for the defective gene, it will remain there. But you expect the healthy gene to produce normal beta globin in the cell. So the workflow or the clinical workflow would be to take hematopoietic stem cells from patients with beta thalassemia. You can do that either by a bone marrow aspiration or by giving GCSF and taking stem cells peripherally, which are known as peripheral blood stem cells. And you isolate those cells and introduce the gene using lentiviral vectors, the normal beta globin gene, to these cells. And you select for the cells that has the normal beta globin gene and then transfuse back to patients with same patient with thalassemia, like an autologous bone marrow transplantation after ablating the bone marrow. So you take stem cells, correct them by putting the normal beta globin gene, knock the bone marrow of the patient, and then put the hematopoietic stem cells back to the patient so that those cells will go and sit in the bone marrow to produce red cells with normal beta globin. So that's the workflow. However, it's not easy as said. First patient with beta thalassemia was treated by gene therapy, treated using gene therapy in 2007. For the last 13 years, approximately 30 to 50 patients have been treated with gene therapy in several 4 to 5 independent clinical trials. The most recent trial, which was again published in New England Journal of Medicine, described 22 patients. 13 of these patients had milder or non-transfusion dependent thalassemia, whereas 9 of those patients had transfusion dependent thalassemia. In 12 out of 13 patients with non-transfusion dependent thalassemia, gene therapy worked. So they were transfusion independent. Whereas only 3 out of 9 patients with transfusion dependent thalassemia, gene therapy had fully worked. And so it's again giving mixed results. However, based on the findings of this trial, marketing approval for gene therapy for clinical use was granted by the European Commission and FDA approval is pending. So gene therapy will be available as a standard treatment even if it has shown some mixed results. Still, there are several challenges that needs to be addressed. So you see there's lack of efficacy in patients with severe phenotype. That's because even though you introduce the normal beta globin gene, that beta globin gene will not express normally if it is not incorporated in the correct area of the genome. We know the beta globin gene is in chromosome number 11, but when you do gene therapy, that beta globin gene can get incorporated into anywhere in your genome. May not be in chromosome 11, may be in X chromosome, Y chromosome, nobody knows. In that location, it will not produce normal amount of beta globin. So the efficacy of production of the normal beta globin is less. So efficiency of gene transfer and efficiency of expression of beta globin is less, and still there will be potential risk. When you do gene therapy, those genes can get inserted in places which are in close proximity to oncogenes, and they might activate an oncogene, and the patient will develop some things like leukemia. And these are done by lentiviruses, and there may be problems associated with these lentiviruses. So still the picture is not very clear with gene therapy, but the results are promising and it will be available very soon as a standard treatment for thalassemia. So what's the difference between gene therapy and genome editing? In genome editing, we can direct or target the area of genome that we are going to mutate. So genome editing, I'll be showing a brief video which will describe the genome editing, which is possibly the most novel technique of gene therapy that is being used. In thalassemia, genome editing is making a specific change to a DNA sequence of a cell, so you can target. Whereas in gene therapy, you put the gene in, you don't know where it gets inserted. Whereas in gene editing, you can exactly target the place that you are going to mutate. Using an enzyme which cuts DNA at a specific place, right? So you can make the two most widely used techniques for genome editing are CRISPR and zinc finger nucleases. CRISPR technique is the most novel technique, and I'll be showing a video how CRISPR technique works in genome editing. Let's try whether this will work.
cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAN. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut, but the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
Right, I hope that was okay. You could hear and see the video. Right, so even with the genome editing, the same workflow for gene therapy would apply. What you want to do is to take hematopoietic stem cells out from patients with beta thalassemia, edit them in vitro, and transfuse them back as an autologous bone marrow or hematopoietic stem cell transplantation. There are 2 approaches that are being tested for beta thalassemia in genome editing. One approach which has progressed to clinical trials is the induction of gamma globin or induction of fetal hemoglobin by mutating the suppressor gene of gamma globin. So the principle here is to, using genome editing, as you saw in the video, mutate a suppressor gene which suppresses gamma globin. So when you mutate that gene, the gamma globin will not be suppressed, so gamma globin will be synthesized. So you will have plenty of hemoglobin F to counteract the absence of hemoglobin A. The other method is, which we have been working on, is to mutate the alpha globin. We don't mutate the alpha globin gene itself, but we mutated an enhancer of alpha globin so that the alpha globin production is reduced in the cells of a patient with beta thalassemia. Therefore, if you reduce the alpha globin production, it will reduce the free excess alpha globin and will consequently reduce the pathophysiology of beta thalassemia. So if we go briefly into two methods, so genome editing of alpha globin suppressor, and scientists in USA and UK have identified a potent gene which is known as BCL11A, which can suppress the gamma globin. So this particular protein is important in suppressing gamma globin after birth. So that is why hemoglobin F production stops after birth because of this particular protein. So what they have done is to mutate this particular gene, BCL11A gene, so that this protein is not produced. So the gamma globin will remain working even in postnatal life. So this has given good results in vitro, and a phase 1-2 trial using CRISPR-Cas9 system that you just saw to mutate this gene is in thalassemia patients is ongoing. So that's one aspect of novel therapies in genome editing. The other aspect that we have been working in collaboration with University of Oxford is to mutate an enhancer of alpha globin. We have identified a specific enhancer of alpha globin, which is located upstream to the alpha globin genes. And this enhancer is quite critical for the expression of alpha globin. When you don't have this enhancer, when you have mutations of this enhancer in natural humans, they will reduce the production of alpha globin. So what we thought is, if we can mutate this enhancer, that will reduce the production of alpha globin, so the alpha globin excess in red blood cells will be less. What we did was this was published in our paper, what we did was to mutate this alpha globin using CRISPR-Cas9 genome editing tools, and then do that in hematopoietic stem cells and grow them in tissue culture plates, and to find out whether it has made any effect. These are the results of that study, where we saw this figure shows the alpha to beta globin ratio in patients with thalassemia and normal controls. In normal individuals, in erythroid cells, alpha to beta globin ratio is 1. Whereas in patients with beta thalassemia, because alpha globin is in excess, the alpha to beta globin ratio in erythroid cells would be much higher than 1. So this bar shows when we mutate the enhancer of alpha globin, as you have just seen, it will bring down the alpha to beta globin ratio to near normal values. So what we propose is, by doing this method, we will be able to reduce the alpha to beta globin imbalance in erythroid cells of patients with beta thalassemia. However, there are several challenges in genome editing of alpha globin enhancer. Although it's been done in vitro in cells, improving it to an efficiency that it works in patients will be a challenge. If we reduce the alpha globin to a much higher value, then there will not be any alpha globin. So achieving a precise balance of alpha to beta globin is also a challenge. However, there are several ongoing research on this, and we will be optimizing the editing of alpha globin enhancer, and we are currently planning phase 1 clinical trials in collaboration with University of Oxford. Now we have come to the end of this session. So in a summary, we just briefly described the current management of thalassemia, and we touched on the blood transfusion therapy and the differences in transfusion therapy for patients with transfusion-dependent thalassemia and non-transfusion dependent thalassemia. specifically for hemoglobin E beta thalassemia. Novel treatment is available for thalassemia. It's not just transfusion and iron chelation. Novel pharmacological treatment that are almost shown some promise are hydroxyurea and luspatercept. Gene therapy in thalassemia have been in clinical trials for more than a decade, but soon that will be approved by the FDA for clinical use. Genome editing is a very new but a powerful tool. The research is still ongoing. Most research is still in vitro. However, some research are now progressing to clinical trials. We don't know the outcome. Hopefully, there may be at some point one of these will work, and we will probably find a sort of a sustained cure with thalassemia. Right. Thank you.